film has a similar mechanistic
Well,
effect.
I just want to say the other thing that's really crucial here is from the medicinal chemistry side, PPIs, protein-protein interactions, which is what we're talking about here, right?
Mm-hmm.
PPIs are some of the hardest things to engineer. Like, this was serendipity. You know, and again, I may be being generous. There are probably some fellow short sellers out there who are like rolling their eyes right now. But like, it's very hard to design a drug that interferes with the protein-protein binding of two separate proteins, right? Typically in pharma, what we're doing is we have an X-ray crystal structure of a specific region, of a specific protein, and we're making this hydrogen bond network to that specific region. And usually it's a receptor pocket or it's another sort of pocket on a protein. I, for one, cannot find the film 1A complete structure. I have like these partial structures. So I'm going to look for a crystal structure for filament A. It may not be possible to create the entire filament A crystal, but thanks to AlphaFold 3, you can actually you can actually render them with some decent fidelity with AlphaFold. So
he tested it initially, I think, with a peptide, which is in one of the papers. It's VAK something. I think that's where he kind of used to narrow down where it was going to bind.
Yeah, and I think it's not surprising that like a very hydrophobic, set of residues on a huge protein would also, bind to another set of very hydrophobic kind of region of A beta, for example. So like it's like, to me, the fact that there is something does not mean that there is something that is working. And, you have to elegantly kind of tie, the preclinical science to some kind of phase 1 PK type science to some phase 2 clinical science. And to me, that story is missing. There isn't this like connection. Now, I mentioned before that I met Remy in 2004. I was an analyst at a firm that his college roommate ran, a major hedge fund. And I was a big fan of Remy. I like Remy a lot. He's a kind of like do-it-yourself entrepreneur. He is not a scientist particularly himself. He went to Oberlin with my old boss, who I still regard very highly, and I think vice versa. I was very disappointed and upset that Remy resigned, because Remy this is his life work. This company is his life's work. Pain Therapeutics almost became very successful. And the fact that he had left the company, or God knows what happened exactly. I wasn't in the boardroom. Maybe you should be on the board, by the way. The fact that he left without an acknowledgement from the company or anything. This is a company that he started 25 years ago, worked day and night, and he's 3 months away, possibly, from the results of his life's work, or at least the latest chapter of his life's work. To me, that is sort of, one, of course, disappointing. I feel terrible for the guy, but it's also very alarming because, if it was just a case of, hey, there's a lot of FUD. I hate that term, by the way. There's a lot of FUD, and we'll see the result when we see the result. There are many companies that have been in this situation. You just stand tall and say, look, we're not going to comment further until the results come out. And by the way, our stock's up, so go fuck yourself, and it's all you have to do. But
the SEC, you had the Dr. Wang indictment. We'll just go with Wang because that's how I usually pronounce it. You had that on June 28th, and then you have, I think it was July 1st, the company announced that they received more information from the SEC, and that the SEC was looking into 2 senior executives. And, the board formed a committee to investigate that additional information. And they said what it was. It basically is around disclosures on a September 2020, I think it's 2020 press release. 2020 press release. And it was about how they disclosed what lab reran the biomarkers, and now we all know it's CUNY, but they said it was a outside academic lab that was blinded for the purpose of reducing the likelihood of bias. And what else did they have in there? They had the results in the CanTab study that was released there had some excluded values, and they said in the press release it wasn't explicitly stated, although it was stated in the presentation and the talk that was done reviewing the press release that day. So yeah, it was around some disclosures there, and that they said they were looking into 2 senior executives, and then after a few weeks, they announced the resignation. And the way if you read the tea leaves, the way it looks like it happened is the internal committee looked into it, and they talked to Remy, and they ended up collectively deciding that Remy and Lindsay resign. But yeah, I agree with you. The thing's only a few months away. I don't fully understand why that was necessary. Or
I actually think it's a little bit worse than that. I think that, and I'm just 100% speculation here. Just having been indicted myself, my fear would be partially. Obviously, I think the board does their process, but one of the problems here could be we're going to see more indictments, which would be terrible, by the way.
The only thing what makes me think that that's not too likely is typically the SEC would attach their claims onto a DOJ indictment.
Yes.
The way I kind of read what happened is the DOJ and the SEC have been talking about this for a long time. The DOJ didn't see any crimes at Cassava. They did see some potential crimes with Dr. Wang's work, and basically they divided it up, and they said, hey, we're not going to press charges against Cassava, so why don't you go full court press on them with whatever you can civilly. We're going to go this route with Dr. Wang. So
yeah, my experience is if they're going to take Dr. Wang to trial, they need witnesses. And the witnesses are Remy and Lindsay. And usually what ends up happening, look at the...
He worked with people in the lab a lot closer.
Well, they're going to put them all in the stand. And
yeah.
in the SBF trial, Sam Bankman-Fried unfortunately had to confront every one of his co-founders and major senior colleagues, except for one, who I should mention. But almost every ex-employee, and some of them he went to college with, some of them he had a long history with, one of them he was romantic with. And it's not uncommon, I doubt any of those people wanted to be there, by the way. It's not uncommon that they simply say things, and I know this from my own case, where they say, oh, are you going to testify against him? Here's the script you have to read. Oh, you don't want to do that? Cool, you're his co-defendant now.
Here's your charge.
Yeah, good luck in prison. And
by the way, your brother's been doing some illegal activities.
Right.
We're going to have to check
Absolutely. So, look, there's nobody that hates the DOJ more than me, and I'd be, just as soon on Remy's side as an individual, but maybe not on his side as this is the great drug for Alzheimer's. So one of the things that every short seller worries about is that when a company has disappointing results, that they try to spin them or delay them or try to avoid reporting them or even
do another trial, plan another trial.
Sure, another trial is fine because that's fine because at the end of the day, Wall Street understands that means it failed. But it's always in the back of a short seller's mind that even if I'm right and the drug doesn't work, what if they say it did? And I think that at least for the short seller side of things, with the DOJ and the SEC, it almost, to some extent, running this company, they're going to be very transparent about what this trial result was. I don't think there's going to be any obfuscation or any spin. It's just going to be here's the p-value, here's the result, here's the result of the primary endpoint. But as you know, even though trials are cut and dry, or they should be, there's things like intent to treat, modified intent to treat, last observation carried forward, best observation carried forward, and you can have like these little discrepancies. But typically a study fails or doesn't. I just want to respond to somebody who says high energy reviews and investments. Martin has been paid by the shorts. Okay, so first of all, I am the short. Nobody needs to pay me. I can.
Well, I thought you said you weren't short.
I'm not. But
okay. You're considering.
Yeah, I'm considering it. And if I do short, there's no short seller who could pay me enough money to buy my opinion. My opinion, the last time I shorted a stock, I made $20 million called Vital Therapies, and I wrote a 40-page paper about it. Before that, I made a similar amount of money in Celadon. I mean, you can look at my track record of public shorts. It's very good in biopharma only. So, I have no need to be paid by anybody. And nor would that be how I would make my money. It's also probably illegal, for what it's worth, to pay somebody to bash a stock. So, I think that
if they overtly say that, that's not what's.
I think you have to disclose if you're being paid.
Yeah.
I'm pretty sure. But
lots of ways to pay somebody.
Yeah, it's nothing that I would even remotely approach because at the end of the day,
I'm not accusing you of that.
No, I know, yeah. But it's just kind of silly, and I just would point that out. So in terms of the mechanism, one of the pieces of homework, because I don't think that we're unlikely to change each other's mind, but you've given me some things to think about. One of the things that I think I'm going to do is I'm going to do a little more research on A beta. It's been a while. I spent many hundreds of hours on A beta in my career. It's a very odd molecule. It's, as you know, it's aberrantly made. There's many A beta species, and they're all slightly different. And what happens to them and why is the subject of a lot of kind of science that's unclear. So I'm going to spend some time on that and maybe make a list of all the things A beta binds to, which I think there's a lot. So I sort of call into question this idea that, it's because it binds alpha 7. Well, what about the other 100 things it binds? Because I do think it binds like a million things. And then do a little more research on filamin, maybe get a crystal structure, like an estimated crystal structure from Google, and see if that can be illuminating and try to predict even the binding event here and where this binding event occurs. Actually, there are binding events that occur that have nothing to do. They're in other words, they're called allosteric. If you're binding on Ecuador, it's not going to change New York. That's typically true, but not always true. And there's
something and not really be causing any chain reaction.
Yeah, exactly. For 2,500 amino acid protein, like it doesn't seem likely to me that binding is going to happen. Now, is this a covalent bond? Is another important question. Because if it's a covalent bond, I'd say generally in chemistry we don't want that. We want hydrogen bonds. But if there's a covalent bond there, that should be sort of easier to spot. I've done a lot of in silico chemistry work, so like I know how to run these systems, do molecular dynamic simulations, etc. So I'll take a look at that a little bit. I'm not going to get any better answers than.
Did you watch like some of the stuff? There's clinical trials that have been run, and so did you watch the Suzanne Hendricks presentation at CTAD?
No.
I'll send that one to you. But she did it kind of goes through. It gets through the end of the CMS study. So it's until 18 months.
So the phase 2 that we were talking about.
Yeah, the open label, and she's a statistician. She does analysis of all the different Alzheimer's drugs, and so she's fairly well known in the space. And in there, she talks about how the treatment group of the Cassava phase 2 open label compared to all kinds of different studies that she has looked at in the past.
Matt, here's what I propose, an intelligent way to, other than me pleading with you that is not valid. Here's maybe a way that maybe you could see where my point is. There are people, would you agree, in biopharma who've made a lot of money investing. They're very good at what they do. Would you say that those people have a skill or a talent? There's people sitting on billions of dollars who have analyzed drugs, and they know, and I'm not talking about Cassava, they know that the perils of open label analysis, and that.
No, there's definitely perils.
Okay, I'm glad you.
It'd be better if it was the big 400 patient blind placebo study. Totally agree.
Right, and all I'd say is that if you went to some of those people, I can think of a couple, Joe Edelman, Perceptive, one of the biggest funds, the Baker brothers. You got a good network, you're a wealthy guy. It would not be impossible for you to meet with the Baker brothers or Joe, and all these people are billionaires, basically. Steve Boyd, Armistice, Rod Wong, RTW, these are just the biggest biotech funds, OrbiMed, et cetera, et cetera. And every single one of them will tell you that when we study biopharma, we don't look at the open label data. We try to avoid it because it tends to just be noise. It tends to misdirect. It doesn't mean it's worthless, but I think you have to be really careful. Yeah.
It can be directional.
Even saying that is, I think, these, again, don't take it from me. Talk to the best of the best, and I think they're all going to tell you you can't rely on this stuff. You can rely on a randomized study, and let's look at that data. It was not statistically significant. That is not good. It didn't.
CMS. It wasn't powered to be.
Wait a second. I don't know if that's.
It literally wasn't. It was
okay,
a 6-month study.
But even if it was powered to be, and the p-value was 0.001, the change between drug and placebo is very small. That's why it's not
because it's a withdrawal study, and if the medicine is working and it's disease modifying, you will have a slower withdrawal than if it is working and it was just symptomatic.
I suppose I can buy that, but I wonder why they would do the study this way, then.
I don't know.
You could do the study like.
I think I would have preferred, if you're going to stick and do open label, just to do the whole thing open label. I don't.
Agreed.
I mean, I don't think that it was particularly useful to do a CMS. And then they rolled those people that were on the CMS back into treatment, and so they have 24-month treatment data on Alzheimer's disease patients. Which, even if you study Alzheimer's disease, any placebo isn't going to perform like that.
I can show you plenty of placebo trials that did. Dimebond.
Not for Alzheimer's, not for 2 years.
Dimebond, yeah, sure. Absolutely. Patients. I've seen it. Dimebond.
Send me info on that one.
Bapineuzumab, Dimebond. There's a host of these, and that's why we just don't trust open label data. It's not meaningful. And, they were still declining in this study. They declined. I'll look at the full data, but it's not like they got arrested development or they started reversing. Placebo declined almost as much as drug. I mean, it was basically the same thing. It was not statistically significant, but I don't even know. I can do a little biostatistics. I'm not even sure what sample size would get you stat sig here, given they didn't give the standard error, but I'm guessing the standard error is probably similar to other standard errors, and I mean, you're going to need, the Cohen's D value would be like 0.2 or something. Like, this is not a effect. And I understand what you're saying on the controlled withdrawal is possibly like one of these things that could putatively distort the rebound and so forth. But if you look at Vertex cystic fibrosis controlled withdrawal study, it is literally a straight line up, then a straight line down when you withdraw it, and a straight line back up when you give it. And I understand CF is not Alzheimer's. The brain is a really unusual thing.
It moves slower.
Yeah, but, if it's going through acetylcholine, then it shouldn't. Acetylcholine is more of just like the tonic underpinning of the nervous system. This isn't like a learning molecule or like a synaptic integrity molecule. So like, if you look at what Aricept does, Aricept works right away. Take it away, you drop right away. It's like, it's not.
Yeah, well,
so if it works like acetylcholine, which is the hypothesis, it should actually be rapid on, rapid off. But
I don't think it's the hypothesis that it works like acetylcholine. I think.
Well, it's a nicotinic receptor, right? Alpha 7.
The hypothesis is that that the neuron is being damaged by that interaction between amyloid beta and the receptors, and the neuron eventually dies.
But the alpha 7 receptor.
Yeah, but that's attached to a neuron.
Well, the neuron's got 50,000 receptors. I mean, it's not going to die because it doesn't have alpha 7.
It's the reaction down through the neuron. That's the MOA is that's actually causing downstream effects. It's not that it's shutting down that receptor. It's that it actually kind of kills the neuron through that receptor.
Yeah.
So it's not asymptomatic. It's.
Well, we hope so.
Yeah.
Yeah, I mean.
So I don't know. I would take a look at the one I just sent you, and then the 24-month data too. So you have 87 mild patients that stayed flat for 4 years or for 2 years, 24 months on the drug.
Well, in 6 months they lost 1 point, so flat is. It's not really flat, right? At 6 months.
Well, it's on the ones that went on placebo were down 1.
1.5.
And the ones that were in the mild group.
No, I mean, we look at all comers, right? We can't look at one group that did better.
Yeah, which one are you looking at?
The full analysis set.
Oh, I see. Like the mild versus moderate.
No, the whole thing.
The whole thing.
Yeah. Yeah. If they knew that mild would be better than moderate, they would have done that ahead of time, right? This is just a like re-examination and saying throw out this data.
Most studies are done mild to early, and so the moderate segment group is a little bit of a risky thing to add to it. But that said, when I talk to the patients that have come off of these studies, there's a number of moderates that have improved radically.
I think I remember when Remy actually was doing an IBS study of naltrexone or naloxone. I forgot it was ultra low dose naltrexone, and I talked to a bunch of patients as well, and they were doing great. This is about 20 years ago, and I learned a very painful lesson about patient testimonials. They're pretty much worthless. I've seen them, and even in cancer, I've seen them to be worthless. Like, it's just hard to, that
Alzheimer's though. Like, there's
I don't think it makes a difference.
There's main symptoms. Like, how does someone that is literally like losing balance and falling down, nothing else has changed in their life except entering a trial, they're diagnosed with Alzheimer's, that they stop losing their balance.
I would say the same thing about Duchenne muscular dystrophy of a dying boy. How could he all of a sudden start walking better in a clinical trial? Yeah. It happens. That when I trusted Remy on a subgroup analysis with patient testimonials last time, I lost millions of dollars. That was 20 years ago. I learned my lesson
the thing is
many times over.
With this, there's about 7 families that I've talked to. Many of them describe very, very similar stories about what's happening. The main symptoms of Alzheimer's.
Is there selection bias though? Is there a selection bias there where you talk to the people doing well, but you're not talking to the people not doing well because they're drooling somewhere and not able to talk to you? So
100%. No, definitely. And because someone that's not responding to the drug isn't going to call me and say, hey, man, right. I would like to continue open label, and they're terminating my program. That's how I ended up interacting with
you're comparing a probability distribution versus another probability distribution. And
definitely.
If you're talking to the right tail of one, you could be talking to the right tail of the other too, and you'll get the same testimonials. It's the mean and the median and the standard deviation that is relevant.
But in Alzheimer's disease, it's not the way the disease functions.
Well, I would just argue at 6 months.
There's not responders.
The data that I have, which is not personal anecdote, but the data that they put out is the drug and the placebo had almost no difference.
Oh, you're just looking at the CMS study.
Yes, at 6 months.
You've got to look at the first 12 months.
Is there any other control data?
There's no more blind placebo data.
So the only control data for this drug does not work. I mean,
the only control data, it's not the mild group showed that it
they should have just done mild then.
Yeah.
I mean, I don't know how you can throw out. It's like saying my team won the Super Bowl in the first half. Don't worry about the second half. It just doesn't.
But
the heuristic just doesn't.
There's 87 patients, and
small study.
Half of them are placebo, half of them are mild, half of them are moderate.
I get that. I just
and the disease, the way this disease is studied, is they're always broken into early, mild, and moderate.
Well, it is and it isn't. Like the primary endpoint can be, for example, for donanemab, for Trailblazer, they did. Let's look at the Trailblazer study real quick.
Tau. I think they used tau. They did mild. They only did early, and they used cohorts based on tau.
Some of many Alzheimer's drugs have tried mild to moderate. It's not like some.
There are some.
I mean, including this one, right? So it's not like this was unusual.
The two that were approved were just early to mild.
Yes, there's been this. You want to treat earlier, you want to treat longer for amyloid beta antibodies. That may make sense for amyloid
Time
beta.
is important. Yeah, that's why the 24-month data is I think very interesting. Is time is as important as quantity.
Time controlled, I would agree, but without a control, I don't know.
Yeah, and that's what we get in the phase 3 study that reads out.
Oh yeah, that's where all the marbles are, right? I mean, that's the big money. It'll either go up 50x or it'll go down 90%, most likely. One of the two.
Yeah, that's why it's a bad short, in my opinion.
Yeah, I disagree, of course. But that's what makes markets, as they say.
It does. It's just your downside risk on it is very large.
If there was a chance that it works, I would agree. I just don't think there's a chance. I don't think there's a chance at all, to be frank. But we have different opinions.
Clinical trial sites have been very positive about it, which you can say what you want. That's anecdotal. The patient families that we've talked to, they describe radical turnarounds. There are super responders to this drug. There are. That doesn't happen in the other Alzheimer's trials. The other Alzheimer's trials, they don't talk about anybody improving. It's just slowing decline. This drug has shown that it can improve, in the clinic.
I think we're not going to convince each other of what is
definitely not.
What is convincing information? Convincing information to me
phase 3 readout
is no controlled results.
I wish we had that.
Well, then we do. It didn't work.
No, you don't. You have a 6-month withdrawal study. 6-month cognition data doesn't mean anything in Alzheimer's disease.
Well, why'd they do it then?
And that's a good question.
No, they did it because they had a hypothesis that it would work, and it didn't work. Like in science, yeah, absolutely. In science, you absolutely, come on, Matt. I've designed these trials myself. You a priori say, my hypothesis is X. You don't just randomly pick. You do a protocol. You write a SAP. The protocol here is available. And they say, we are designing this patient population to target this change in ADAS-Cog and this separation at 6 months. And this is how much power we have to see this much change.
It was informational. They did not design that study to show statistical significance.
There's a protocol here, right? Let me.
I don't know for the CMS. That wasn't the goal of the study.
Yeah, go ahead. Sorry.
If it did, if it honestly, I would have sold some of the stock when those results came out. If it was statistically significant in that 6 months, because it would have showed that it was symptomatic and it was maybe more like Aricept. So the fact that it slowly declined, there was some separation, but not rapid separation. I look at it as a positive. That's what I would want to see.
I understand. I mean, since we're recorded.
I wish we had a longer study, and I've got to run soon because I got to go pick up my kids from school. But if it were a longer study, then I think it could have gotten it. But 155 patients for 6 months in Alzheimer's, it's not Obviously, it's not a statistically significant design.
Diamond Bond did it. Diamond Bond got phase. So did Trailblazer. I mean, it is possible. Diamond Bond was
not with 155 patients.
Yeah, Diamond Bond did it with around that much. I think that
not 6 months.
Yeah, you got to look at the Diamond Bond results. It was published in Lancet. This isn't even by the way, this isn't even published, as you know.
Yeah, I'll look at the Diamond Bond one. I'm sure the people that I work with on this stuff have.
Yeah, we're on the YouTube, so I would just say that I implore you. I'm sure this will be cut and taped either way, whatever happens here. You're right or I'm right. I implore you to save your money. Do not waste your money here. You're going to lose the money. That's just my 2 cents. I know your opinion is you might make $1 billion in this trade or more. And you know, for your own sake, I hope you do. You sound like a nice guy.
And Alzheimer's patients, but yeah.
Yeah, no, I want Alzheimer's patients to win too.
And I'm serious, I'll shave my head with you. Maybe not together, but...
Yeah, I don't think I'm going to be losing these beautiful locks, but I think let me do some more homework and we maybe can have another discussion.
Yeah, I'm game.
Thank you so much, Matt.
Thanks for the talk. See you later.
You too, bye. So that's how you have a conversation with an adult. Some of you guys out there are not adults. You're little ants. And you know what I do to little ants? I squish them like this. Squish, squish. Squish, squish. Don't be an ant. Jokes aside, I think bug people. Look, I think that I think there's no chance at all that this drug could possibly work. I think that Matt, I mean, look, he's very smart and successful in his own field. This is my field. Nobody looks at data other than controlled trials. You do not do a trial for fun. It's really expensive. You do a trial hoping you prove something. When you don't prove something, you really have to start over. I would do another 6-month study. I wouldn't do a phase 3 study based on this data. That's kind of an act of desperation, I think.